What's up, Genetics 311? This is my exam mini video 4, and we're going to be talking about prokaryotic DNA replication. Got that circular DNA there. All right, so game plan is we're going to draw out the process of DNA replication in prokaryotes, and as I get to each step of the process, I'm going to be introducing new terms and bringing in new items to the drawing, and I will describe each part, what it means, and what it's doing in the process and its significance. As stated before, the diagram is going to fill up with more ' as the process goes on, but for now we're going to start with the two DNA strands being unwinded by the helicase, having the 3' to 5' strand on the top and the 5' to 3' on the bottom. And the gyrase is at the front, kind of relieving the pressure and ensuring that no supercoiling happens. And we have those phosphodiester bonds that are keeping the sugar phosphate backbone, the structure of the DNA in place. All right, we have a few more items up on there now. So we have the leading strand defined, as well as the lagging strand, where the leading strand will have a continuous replication and the lagging strand will have the Okazaki fragments, and they're represented by the green segments between the primers, which are made by the RNA primase. And the leading strand will go away from the point of origin of replication, while the lagging strand will actually build towards the origin of replication. Also, I'd like to add a quick note that the single-stranded binding proteins are put in place to make sure that rewinding does not happen. Alrighty, now we have the big team players added into the process. We have DNA polymerase 3 and 1. DNA polymerase 3 on the leading strand will go ahead and add all of the nucleotides going in the direction of replication, and then on the lagging strand, it'll go towards the point of origin, and the DNA polymerase 1 will replace the primer nucleotides in each. Finally, we have ligase represented by the black circles, and their job is to connect and seal off the Okazaki fragment. Okay, now we're going to talk about what would happen if DNA polymerase 1 could not perform its 5' to 3' exonuclease function, or just if DNA polymerase 1 did not work. Essentially what would happen is the DNA polymerase 3 will go in and add the wrong nucleotides, and the RNA primers will not match up in the DNA strands. That is all for this exam video. Thank you very much. Hope you enjoyed.